Good afternoon, everyone.
Good afternoon, sir.
Yeah, good afternoon. Okay, we'll get started in 2 minutes. Okay, let's get started. Okay, so yesterday we were discussing about the first concept of, I think we were talking about one aspect here with respect to the population genetics. I think covered factors there, and also we discussed about the few components. I've sent you the content related to that, right? So I'm not sure if you happen to read anything from there, like Bradford study I've given you yesterday. So these type of case studies are important. Okay. So Bradford study briefly talks about an ongoing study. It is not just because I gave one article, but this is still an ongoing study, huge study. And as per this, there are a few thousands of babies are being monitored for any abnormalities that are happening because of consanguineous marriages. Okay. So born in Bradford project, it is called BIB. Even you can go to the actual website of this project. If you type born in Bradford project, you will see it, but there are many dimensions to this project. So instead of giving you the link for the actual project, what I have done, I've given you an article that was published in BBC in 2013. Okay. So this project briefly talks about how people from particular religion, that is in this particular case is from Pakistan, have seen increased number of certain recessive disorders in them. That is because of the consanguinity that they follow. So marrying the cousins resulted in higher number of homozygous condition, higher number of homozygosity there. Okay, so these type of projects, you can use them as case study. This is a very well-known project. So for consanguineous marriages, as I was telling you, if you have also posted an article, okay, you don't have to read everything in that article. When I post something like this, I would specify what is important, because these articles are not written for our anthropology courses, correct? So, but we want some sort of reference to rely on to write. So the review I've sent you, this was published in 2012. So this is mainly meant for how in a particular place they are working on families, who are into consanguineous marriages, how to counsel them. So in the process of explaining in this article, they have briefly talked about what is consanguinity, what is the definition of consanguinity. Some of these points I've already discussed in the class. This is a source I was using for that. So inbreeding coefficient component was actually taken from here. And you can also see the global spread of consanguinity matings or consanguinity marriages. Okay, African regions, for example, or Asia, we have seen that. And categories or types of consanguineous marriages also given here. First cousin marriages, uncle niece marriages, second cousin marriages. Okay, so if they ask you for the types, briefly you have to mention about this. And overall trend of consanguinity, we have seen from one particular source how consanguinity varies in religion to religion. We have some data I have shown you there. I forgot to mention you the reference for that. I will send you that. I didn't actually completely forgot about that. I'm going to send you the reference for that. Otherwise, this global consanguinity trends you can see from this article. And also they have nicely organized the information here. Why are consanguineous marriages are favored? What are the benefits of consanguineous marriages? Okay, and the effects of consanguineous marriages, health issues. And here it briefly talked about the counseling aspects. Okay, actually in the 9.4, we're also going to discuss about counseling, role of counseling as part of anthropology, physical anthropology, in dealing with certain genetic problems. Okay, so there I'm going to help you briefly understand this. For now, just see how consanguineous marriages can be studied here. Various dimensions are given in the review, so you don't have to focus on the second part of it. Once we finish talking about counseling, if you're interested, you can see how counseling is planned. For this, from this review, I shared with you mostly the first part. Okay, what is consanguinity, types of consanguineous marriages, why are they favored, what are the issues, genetic issues of them. Okay, if you focus on that part, as well as I gave you idea about what are the effects of consanguineous marriages. If you focus on that, that would be sufficient for our anthropology. Okay, right. So there are other topics from this unit where questions are sometimes asked. Today, first we are going to start with genetic load. Okay, there is a topic called genetic load, and then we'll move on to genetic polymorphism. Okay, first we'll begin with the genetic load part for this. So what is genetic load? So in literal sense, load means what? Load means burden, correct? So what is genetic burden and what causes it? Okay, and in the past exams, I've also seen they asked once about calculating how is genetic load in a population is calculated. So we are going to focus on these three things. Our goal is first of all to understand what is genetic load, okay, what causes genetic load, or what are the different types of genetic load that we are going to talk about, and then how is genetic load calculated. Okay, based on the questions that are asked in the past, I've designed information like this. Otherwise, there are many dimensions we can go with this, but instead we're focusing on that component. Okay, genetic load. I think it's a very bad timing for me, so I lose power here. Thankfully, my internet works. Right, so let me share this screen with you. Okay, load is not here. Where is my PDF? Well, polymorphism, genetic polymorphism, genetic polymorphism, notes. Okay. My God. Okay. Combination load. Okay, I'm sharing the screen. So this is simple concept. Okay, we already discussed some of the aspects related to it, so it won't be too difficult for you to understand this. So as I briefly told you, load means burden. So genetic load means so any change that is happening in the DNA of a population, DNA of the individuals in a population, which decreases its fitness. Correct. Yesterday we talked about, as part of natural selection, we discussed about fitness, right? So reproductive fitness, survival fitness, we talked about. So any changes that happen in the genome of humans, okay, for that it could be any organism, any species, but for us we are mostly focusing on humans. So any such changes, they can affect fitness positively or negatively, correct? And selection acts on those that are beneficial to us, they are maintained. Those that are not beneficial or those that are actually harmful are eliminated from the population, correct? But because of those negative alleles, or those alleles that can actually have a negative impact on our survival, can result in some amount of load. So the negative impact of changes in genetic sequences lead to genetic load, or it is referred to as genetic load. I hope you are following this. You will understand it with the examples and types I'm talking about here. So it could be considered as reduction in the average fitness of a population. Because of a change in the DNA, if the fitness of that population has decreased, such a change is called genetic load. Such a change is called genetic burden. Let's say that there is a couple of people in a population living in a given area. A mutation has taken place. The mutation has a negative impact on their survival. I know it normally gets eliminated over a period of time, but even in the process of elimination, these populations actually experience some negative impact. That is called genetic load. Okay. So reduction in the average fitness of a population, and it's a relative term because we don't know what is best, correct? So when it is compared to the population with optimal genotypes, so it's relative. When you compare it with the best case scenario, simply I'm just giving an example for you to start thinking about it. The example here, let's say that O blood group is really beneficial. This is just a theoretical situation. Let's say originally people might have had O blood group. Later, because of a mutation in the gene coding for this antigen on RBC, it resulted in A blood group. But for some reason, A blood group is not as good as O blood group in terms of providing survivability to those people. Then what do you say? The change in that blood group related antigen has resulted in a burden, because now they are carrying a version that has changed from the O blood group, which is actually causing burden. It is affecting their fitness, or it is reducing their fitness. That is known as genetic load. This is a very theoretical situation that I just wanted to explain it to you. O blood group is optimal, A is not optimal. In that case, A is causing burden. Change in that particular gene resulting in A is causing burden. That phenomena is called genetic load. Okay, are you able to understand this statement now? Reduction in the mean fitness or average fitness of a population relative to a population composed entirely of individuals having optimal genotypes. So compared to the optimal genotype, any change that happened to that, if it has a negative impact on that population, it is known as genetic load. Okay, any difficulty in following this? There is one condition where particular gene sequence is useful, optimal, but that sequence has changed because of mutation or because of other factors. So what happened? That fitness is changed. If fitness has reduced, then you refer it to that process as genetic load because it is causing burden. It is reducing the burden. Such a change in that DNA sequence of a population is called genetic load. Okay. If you're stuck in understanding this concept, feel free to ask me questions on it.
So one case if we take, so if there is a negative, so the whatever that mutation is there, it's not beneficial for that population. But if that population migrates to some other place, and then it won't be called a genetic load, right?
Yes. Yeah. Then that it won't become a burden to them. If the same mutation is useful or beneficial to them in another region, another environment, it won't be considered as genetic load. It has to negatively impact their survival.
Genetic load is with respect to the environment.
Genetic load, yes, correct. Genetic load can be considered with respect to environment, but you may find a few such changes which are beneficial to in some environment, non-beneficial to other environment. But definitely you will have cases. In that sense, it won't be considered as genetic load. So it is environmental related. Okay. But I'll tell you the idea where this had come. Okay. Before getting into the other details. So if you got now idea about what could be genetic load, DNA changes, but is a mutation only way to cause change? No, mutation is one key source for adding change to the population. Okay, but there are other ways where changes can happen. They can also result in genetic load. And how did this concept actually originated? So people, when they started learning about how inheritance happen and how certain changes in DNA are detrimental to us, that's when they thought about genetic load. In the handout I'm going to share with you, okay, I have given a brief introduction part to this. Okay. So the basic idea of genetic load was first discussed by Halden in 1937. So this concept had begun in 1930s. So where Halden, he wrote something called the effect of variation on fitness. He was discussing this concept where he looked into how variation or change in DNA sequence affects fitness. When he was studying this, he came up with the idea of genetic load. And he showed that decrease of fitness in a species is a result of recurrent mutation. Recurrent mutation means a mutation that keeps happening. Because imagine if one particular person receives a mutation, right? So what happens? He may inherit that to the next population. Eventually, that mutant is detrimental. So either it gets eliminated or at least for some time, for some duration, it gets maintained. Correct? So what happens if this gets maintained? You notice that burden on the population. And this mutation may not happen only in one individual. Sometimes recurrent. It does happen, keep happening in other individuals and repeats in the generations, even if it is actually taken out. So selection takes out the mutation, but the mutation may reappear. Maybe we keep exposing ourselves to chemical pollution, and that chemical pollution may result in the same mutation. So those recurrent mutations can keep affecting that population. So this is what Haldane studied as part of his genetic studies. Okay, later Muller, another famous geneticist, Muller in 1950s also observed the same concept independently. So both Haldane and Muller can be credited with respect to the genetic load. And in the process of understanding how changes in our DNA affect our survival, they came across this concept. Okay, does it make sense logically? Like why and when people think about this type of concept, they notice the change and they're checking how this change is affecting the survival of a given population. Okay. So it looks like as per the literature, estimations, okay, you cannot exactly count these numbers, right? But as per the estimations of this phenomena, that every person is carrying an average 3 to 5 recessive alleles. So on average, all of us seems to carry about 3 to 5 alleles in us, which in homozygous condition can be detrimental to us, can cause death or can cause disability. So it can, we compromise when we have this type of alleles in us, we do compromise for the survival. So on average, we may carry about 3 to 5 such alleles. So inbreeding or consanguineous marriages we talked about yesterday, they can result in that detrimental effect on the future generations. So it is not very unusual. We all do carry this, but only in combination of another such mutant, it would affect the individual. Okay, so this is a brief idea about what is genetic load. Okay, there are some ideas here, but that will be a little too much for you now. So I'll let you read it, and if you are confused, I will explain it later. There are a few theoretical concepts that are given here. But first, I would like to talk to you about different categories of genetic load. Okay, and how do we calculate genetic load? Then we'll see how to deal with this information. Okay, so what are the different types of genetic load? There are many different kinds of genetic load. Some of them you can easily think of, right? Assume that we're talking about variation that is causing the, that is reducing the fitness. So what is the source for variation? Where is variation coming from? Some of these things we talked about in evolution as well as in Hardy-Weinberg equilibrium. Where is variation coming from? What are the main sources for variation?
Mutation.
Sorry?
Mutation.
Mutation, correct. Mutation, is a mutation only source for variation?
Genetic drift, gene flow.
Correct. So drift, but drift is normally happens in a context of small populations due to random chances. Correct. So recombination, when two people married, recombination results in new combinations of alleles. Okay. Uh, what else? So recombination, mutation can be seen. There are a few other things which are not evident to us, but I'll explain you in what sense they can be connected to genetic load. Okay. So first we'll begin with mutation load. So there's nothing much we learn here. We only learn of how mutation can reduce the fitness of a population. And in what sense something like that can be called as a mutation load. Okay. So genetic load created by mutations that introduce either inferior or superior alleles to the population. So any mutation, okay, usually the mutation that affects the reproducibility, not reproductive ability, that affects the reproductive ability. Second, something that actually even reduces fitness, okay, and can cause death. Such mutation leads to mutational load. So load happening because of mutation is called mutational load. So nothing complicated. Here the load is happening because of the mutation, sudden heritable change happening in the DNA sequences. Such a load is called mutational load. Yesterday we were talking about mutations can either be positive, negative, or neutral. Correct? Here mostly we are focusing on those that are negative. They introduce some alleles which can affect the survivability and which can reduce the fitness of a population. Such a load is called mutational load. Is that clear? So what is mutational load? Okay, I have given this in the handout. So it is a reduction in the fitness caused by accumulation of deleterious mutations or negative effect of mutations. And fully recessive mutations are maintained in higher frequencies than partially recessive ones. Okay, and they cause the reduction in the fitness, mean fitness of the population. Okay. So such a load is called a mutational load. Next, I would like to tell you briefly about segregation load. I'm using a different order here. Okay, so I'll pick something that is easier for you to follow and later a bit more difficult ones. So what is segregation load? Where did we hear about segregation? Where did you hear the term segregation before?
Law of segregation. Mendelian
Law of segregation, right? Mendelian law of segregation. Segregation means
separation.
separation. Okay, so let's say that parent A contains one normal allele, one mutant allele, because we have two copies of alleles, right, for every gene. So there is normal and mutant allele in that person. But when gametes are formed, we will have one gamete carrying the normal allele, another gamete carrying the recessive allele or mutant allele, right? So segregation, because it has equal possibility of segregating normal allele and mutant allele. Can that result in a load? So here it is in the cause here for carrying the gamete that carries a mutant allele, it is a result of segregation process. It is a result of segregation process. So that mutant, if it also joins with another such a same product, such a mutant variety, it would result in the diseased condition, right? Thereby reducing the fitness of that population. So segregation load is what? The load that is caused when a population is segregating less fit homozygotes. So what happened is homozygotes I'm talking about, it's segregated, one mutant, another mutant. So two recessive alleles are together. So this is homozygous condition. And this is a result of segregation. Okay, so this is one type of genetic load where we are blaming segregation to be the cause of this, which is a natural process. So sometimes you may question, so segregation happens, anyway has to happen. But in literature, some people have thought and explained this as a specific type of a load. And here the new combination is formed beginning with segregation. Such a load is called segregational load, where segregation of less fit individuals. Here, homozygotes. Heterozygotes are better. Today we are going to talk about balanced polymorphism. In balanced polymorphism, sometimes we have an advantage to have one normal allele, one mutant allele. But two mutant alleles will cause effect, homozygous recessive condition. And when homozygous recessive conditions are happening in a given population because of segregation, such a load is called segregational load. Okay, these ideas are a bit theoretical. We have to use the concepts that we learned in different classes. So mutation is straightforward, a mutation that reduces the fitness of the person and the population is called a mutational load. Segregation, the segregation process where recessive alleles come together. Recessive alleles are separated and the next step, they join other combination, thereby resulting in the less or inferior types, reducing fitness. Such a load is called segregational load. Okay, segregational load. Next, recombination load. What is recombination? And where does it happen? So what is recombination load and where does it happen?
Mitosis.
So what is recombination first? So for many of these things, you know what? You have to remember, starting with the general concept. Look at the term recombination. Re means again creating combinations. Combinations means joining things from different sources, combining them. Okay. So recombination process takes place during cell division. It can also take place during mitosis, but our focus is usually on meiosis. It does happen during mitosis as well, but we are interested more about recombination that is happening in meiosis. Why? Because That is where next generations are influenced. Mitosis would only affect an individual, not the next generation. So recombination is a process of creating new combinations where before alleles are formed, before gametes are formed, what do? You actually pair homologous. You pair homologous chromosomes, similar chromosomes, chromosome 1 from mother, chromosome 1 from father, for example, and they exchange genetic information. So what do you give to the next generation? Combination of both of your parents. So this is a recombination process, right? But what if recombination creates a variety that is inferior, that reduces fitness? Can you call it recombination load? The load that is created because of recombination process. Okay, that is called recombination load. Combinations of alleles that have evolved to work well together may not work when recombined with different allele. Okay, this is just a little complex way of explaining what I just said. So those alleles, let's say the parent generation, were working great, were actually not affecting the fitness. But when it's segregated, and segregated is the first step, and then recombined with a new form, in this new combination, it is affecting the survival. Then what happens? You can call that recombination. So, okay, so here, sometimes the deleterious alleles that are harmful to us, okay, can combine. Okay, if you can digest, we have not talked about this information in previous classes. There's something called epistasis, synergistic epistasis. This is a bit complicated topic, but term. But briefly, what does it mean? Sometimes, it is not a particular gene acting directly. It is, interplay between multiple genes. So one masking the effect of the other. That happens. So sometimes what happens, deleterious alleles, when they put into a combination, now this combination is greater than the predicted from considering them in isolation. So those alleles in a previous generation were not causing harm. In that combination, they were not causing harm. But in new combination, when they came together with the, together with the alleles or the genes from another individual, now in this combination, they're causing problem. And this is all happening because of the recombination process. At one stage, it is happening because of segregational load, because mutant variety is being placed into a gamete. Next, recombination, what happens? The result of recombination, A and B, two parents, their genes got exchanged. Then what happens? A new combination compared to their previous generation combination. Whatever your parents had, whatever your mother had, whatever father had, didn't have a problem. But in your generation, this new combination of alleles are causing a problem. They're reducing the fitness. But of course, we are not studying at the individual level, but this is happening at the population level. Such a load is called recombination load. Are you following this? Are you able to understand this? So just here, idea is very little superficial here. So what is causing the load? Mutation causing the load, mutation load, and segregation causing the load, segregational load, recombination causing the load, recombination. So load can happen, genetic load, genetic burden can happen due to many of these events. Okay, the next one is migrational load. Now you can guess. People who traveled, moved to new region, they carried some deleterious alleles in them, and they actually mated with the people in that new population. They passed down their mutant alleles to them. So this is happening because they migrated to that region. Such a load is called migrational load. Result of non-native organisms, people who don't belong to that population. So let's say some people traveled from, I think we're talking about dark skin, fair skin in the last class. Similarly, let's say people from Africa traveled to America. Those skin color genes are not useful to Americans. So non-native people move to those regions who are not adapted to that particular environment, and they move to another environment. It is opposite to what you guys were talking about, right? If you move to another environment, would that become a load? It may not become a load, but opposite is true here. So people traveled from one region to another region. The whatever adaptations that they have is not useful to new environment, not suitable to the new environment. So what happens when they marry someone from Americas? What happens? Their children may carry genes that are not favorable to them, are not suitable to them. But why did they get into that population? Because they migrated. So such a load is called migrational load. If they breed with individuals who are adapted to that environment, their offspring will not be as fit as they would have been if both of their parents had been adapted. Such a load is called migrational load.
Sir, one question.
Good.
Can we have multiple loads at the same time also?
Yeah, of course. Migration, segregation, recombination, they do happen. So there can be multiple types of loads existing at the same time. Because all these events are doesn't happen individually. I mean, they are always happening. They're simultaneously happening. So load, all different types of load can be present at a given point of time.
Sir, isn't the segregation load responsible for all the other loads, because that is what ultimately is happening?
Yes, if we consider yes or no, okay, if we consider segregation is a result of everything, but assume this. So segregation, what does it do? Segregation separates that mutant allele and creates a gamete with that mutant allele. How is it different from recombinational load? How is it different from migrational load? So first migration load, let me take migration load. Without even migration, segregation load can happen, correct? Within that population also, mutant allele can, because of segregation, can result in the new combination, result in the gamete carrying the mutant allele. Doesn't matter whether it's migration or not. But to differentiate it from migrational load, you can say the segregation has a potential because they migrated. So in that sense, it is more migrational load than a segregational load. Wouldn't have that situation where no migration has happened, but segregation has taken place. So to differentiate that, they would talk about segregational load separately from the migrational. This is one explanation. Second explanation is recombination. So when does recombination happen? Before or after gamete formation?
After.
So recombination takes place after gamete formation. Okay, it could be before or after. It's like chicken or egg-like question. Correct? So assume that one generation, I'll give you the context here. Parent generation. Parents have formed their gametes. Sperms and eggs are formed. So if gametes are formed, has a recombination already taken place, or recombination will take place after gamete formation in that generation?
After.
After. Okay. What happens in recombination? We just talked about this, right? I know many students have difficulty in understanding this concept. So gametes are formed as a result of what process?
Meiosis.
Meiosis. And where does the recombination take place in meiosis?
Meiosis 1.
Meiosis 1, stage 1. So in stage 1, are the gametes already formed? No. Gametes are formed at the end of meiosis. So recombination happens first, then gametes are formed. After new combinations are decided or defined, that's when gamete receives the new DNA, new combination of DNA. So where did this happen? This process of recombination has happened earlier, even before gamete is formed, even before segregation has taken place. But why are you considering as recombination load? Because the problem is not because the segregation has happened, because the new combination is formed when two parents with one normal and recessive allele have come together, or two recessive alleles have come together. The new combination is causing the problem. So recombination load is different from segregational load. And segregational load is different from migrational load. Okay, they are connected because segregation has to happen in every generation. So you can say everything is segregational load. But is the problem happened because of segregation? It mainly might have happened because of migration, because of recombination. Okay, is that clear, Sunil? Is that making sense?
Yes.
Okay, so migrational load, recombinational load. and segregational load, mutational load we talked about. Few more loads. Okay, you don't have to explain all of them in the exam, okay, depending on what is the demand of the question. Few of these types. Remaining you can mention. Most importantly, mutational load, segregational, recombinational load you can talk about. There are many ways that you can define load. So there are few of them that I'm briefly going to mention to you. I think we'll skip balanced load because it's just an idea which already ties up with segregation and recombination. So I think I'll talk to you about incompatibility load. Incompatibility load. So you all know about Rh blood groups, right? Rhesus factor, Rh blood groups. Are you familiar about? You all know this, but do you know what is Rh? So blood groups, A positive, B positive, A negative, B negative, A, B, O, correct? That positive and negative refers to what? Another factor, another antigen. This is rhesus factor. Okay, so Rh factor means, I think I briefly talked to you about this in another class. So we have O blood group, there are no antigens, no specific antigens. There's H antigen, but usually there are no antigens in a general level. A antigen, B antigen is present, correct? But in addition to A, B antigen, sometimes we also have, usually we have another antigen called D antigen, or it is also known as Rh factor or rhesus factor. So if a person carries Rh factor and A antigen, what is his blood group? The person has A antigen on their RBC plus Rh factor. What is his blood group?
Negative.
Negative. Okay. So what am I saying here? I'm saying that A antigen is present on their RBC and also Rh factor is present. If Rh factor is present, you say it is positive for Rh factor because it is present. If no Rh factor antigen, no D antigen is present, you call it negative. So A negative. A person with, let's say I'm drawing a circle, on top of it I'm drawing A. A means A antigen plus Rh antigen. The blood group is A positive. Positive refers to positive for Rh antigen. If Rh antigen is present, it is positive. So A positive. Same thing, a person with B antigen and Rh factor antigen present on his RBC. What is the blood group?
B positive.
B positive. Exactly right. So next, no antigen, no A, no B, only Rh factor. What is the blood group?
O positive.
O positive, exactly. None of these antigens are present. No A, no B, no Rh. Shweta?
So O blood group.
Positive or negative?
So positive.
No Rh factor. No A, no B, no Rh factor.
So positive.
What is positive? Positive means presence of something. What is present here? Nothing. No
Nothing.
A, no B, no Rh. So all negatives. So it's O negative. Okay. If there is Rh antigen, it is positive for that. So A positive means A antigen, Rh antigen. B positive means B antigen, Rh antigen. All negatives. No A, no B, no Rh. It is O negative. So Rh factor is the third antigen, D antigen. So presence of it defines whether a person is positive or negative for Rh factor. All positives, A positive, B positive, O positive, AB positive, all carry Rh factor along with the other antigens. Okay.
So what is the importance of that RH factor?
What is the importance of?
That RH factor.
Importance of Rh factor. So I don't know whether we specifically know why we have it, but we know what we can do with it. Okay, the importance is if there is no match, let's say a person with B negative blood group met with an accident, he needs blood. So we check compatibility there. A person with B negative can take blood from what blood group people?
B negative, O,
So B negative person can give, or
O,
O
O
negative.
negative. O negative can give. Can O positive can give blood to them? Can O positive person donate blood to B negative person? Sorry, B positive person. O negative, B positive. No. The idea is this. Because if you say O, if you have a person who is O positive donating blood to the B negative person, let's say this scenario, this is easier to understand. The receiver is B negative, the donor is O positive. So what happens? This person is introducing, O positive is a donor, he is introducing what antigen? There is no antigen, no B antigen, so he is O, but there is Rh factor. So you are introducing Rh factor into a person who is B negative. B negative means he's a Rh factor in his blood. The B negative person, do they contain Rh factor? No. So if you're introducing a new antigen into his body, what does his immune system do? It will react to it. It makes antibodies. So it will destroy all the RBCs that are given to them. So they're not compatible. It's incompatible. So Rh factor and idea about AB blood groups is important in checking the compatibility. So we'll take blood from whom the body can accept it. So O negative people are universal donors. We take blood from them. Or he is a B positive person, he can take it from O positive. But a B negative person cannot take the blood from O positive person because O positive person carries Rh factor. When you donate the blood to the person with B negative, his body, receiver's body will recognize the Rh factor and will make antibodies against it. So these information knowledge about blood groups, most importantly, are useful for compatibility, saving people's lives. But we also have other advantages, like we are going to talk about these applications. Paternity disputes. You can actually understand paternity to such an extent using blood groups. There are benefits. We have genetic markers separately in 9.6. There we are going to learn a few of these aspects, and partly in Unit 12. Okay, we'll talk about AB of the groups and their uses. But for now, our focus is this. How can this be a load? How can this become a load? Okay, so let's focus on what we have been discussing. How can Rh factor become a problem or incompatibility load? Okay. So think about it. A mother, okay, who is conceived and is about to give birth to a baby, okay, whose blood group is Rh negative. So let's say her blood group is B negative. But her husband either is B positive or A positive. So I'm repeating this. The mother is Rh negative. Mother is Rh negative. Father is Rh positive. Meaning father contains Rh factor, mother doesn't contain Rh factor. So she's Rh negative. Is it possible for them to have a baby who is Rh positive? We'll work on this. Okay, we'll work on these combinations and calculations in a different class. But for now, just focus. Just theoretically tell me, if father is Rh positive, mother is Rh negative, can baby be Rh positive? Is it possible to have a Rh positive baby?
Yes, sir.
Yes. Okay. Everybody agrees on this? If you're unsure about whether it is true or not, do let me know.
It can depend on if Rh is dominant or recessive.
Okay, Rh is dominant. Okay, and let me tell you, it is dominant. Because see, presence of something is dominant over absence of something. Either you have it or you don't have it. So it's a simplistic scenario. So it is having it is dominant. So yes, there is a possibility. It is also possible for this couple to have children with No Rh factor, that is also possible. Okay, so for that we need to work on some permutations and combinations. But for now, believe me that Rh positive father and Rh negative mother can give rise to Rh positive baby. But what if baby is Rh positive but mother is Rh negative? Mother is Rh negative but baby is Rh positive. What happens? Will there be fight? Not in that particular, what is that? Not in that particular birth. But later what happens from the baby, especially when baby is during the delivery process, what happens? Baby's blood can be mixed with mother's blood. Baby's blood can be mixed with mother's blood. So what happens? Mother got exposed to baby's antigen. Mother got exposed to baby's antigen. Excuse me. So mother gets exposed to the baby's antigen. Then what happens? The same fight I was telling you. Mother received some amount of blood from the baby. So her body reacts to it. But nothing happens to mother. Nothing happens to mother. Okay. Nicely explained in this image here. Rh positive father, Rh negative mother. Rh negative mother with Rh positive baby is possible. And Rh positive baby's blood cells enter mother's blood. So this can enter mother's blood. How does mother's body reacts to it? It will make antibodies against the baby's blood. It will make antibodies against the baby's blood. Would that kill the baby? Would that kill the baby? If she makes antibodies against the baby's blood, would that kill the baby? Logical, very logical. So mother had a baby. Baby has Rh positive blood. Mother is Rh negative. So mother can be B negative. Baby can be B positive. Let's see. So when baby's blood got into mother's blood, what happens? Mother's immune system will make antibodies against you. Would that kill the baby? Would that kill the baby?
Since for the first baby, maybe blood is entering at the delivery process, exposure time would be probably less. So it may not affect her much. But during the second baby, since it will stay longer in the mother's womb, so the exposure time is a lot more. So there is a chance that they react and may affect the baby or worse kill it.
Yeah. I think your idea is more or less correct, but little correction is needed. Yes, but I agree with you. I think your idea is correct. Yes, others. Do you agree with that? Did you understand this explanation? What Vishal just said? Okay, let me explain that. So, baby is already born. Baby is delivered from the mother's womb. And no blood is given from mother to baby at that time, correct? Baby only receives the blood, nutrition, oxygen from the mother when the baby is in mother's womb. But baby is already delivered. Only during the delivery process, this mixing of blood has happened. So baby is not affected in the first process, first pregnancy. But the second pregnancy, this can cause problem. Why? Because this mother already got exposed to the baby's blood. Her body made antibodies against the Rh positive, Rh factor. If second baby happens to be Rh positive baby again, if second baby, later pregnancy with Rh positive baby, if that is Rh positive, if she's already, the baby is there in the mother's womb, just conceived, baby is growing, mother's blood will now carry antibodies to that baby. Then the problem will begin. Then the baby's blood will be attacked by the antibodies present in the mother's blood. That is when problems happen. Rh antibodies attack the Rh positive baby's blood cells, causing Rh disease.
Sir, here in this, so baby also receives blood from mother in the womb.
Who?
The baby also receives blood from the mother in the womb itself.
Correct. Yes, that is how baby is fed. That is how baby is given oxygen. Correct. So mother to the baby connection is this. Baby stays in the mother's womb. We'll talk about this in unit 10. I'll describe you with images there. So there is a connection between the mother and the baby, correct? Placenta. The mother's side, umbilical cord from the baby is connected. So there is a pipe going from mother to the baby through blood vessels, through blood, food is given, oxygen is given to the baby. Right? But the problem is happening here after the baby is born. So first baby is not affected. But when second pregnancy, when mother carries his antibodies through her blood, these antibodies reach the baby and Rh factor containing RBCs are attacked. So thereby killing the baby. This is called incompatibility load. What is the problem here? There is no compatibility between the mother's blood and the baby's blood. So this is called incompatibility load. How is it load? Because it is affecting the baby's survival. And what's causing it? Incompatibility in the blood groups. So this is called incompatibility load. This is more severe. This is more severe, but this can happen with any blood group, A, B, but normally that is less severe. So usually we study about Rh positive incompatibility, Rh blood group incompatibility, because it can cause death of the baby in mother's womb. Yes.
Sir, does it mean, whatever period the baby is in the womb, that is the time required to develop the antibodies in the mother?
No, antibodies are made between the pregnancies. So when she delivered the first baby, that is when her body got exposed to it. Her blood starting to make antibodies. Okay, so that happens within few weeks after the baby is delivered. So antibodies are there. It will remain for a few months, few years sometimes. Okay, depending on the type of antibodies. So these antibodies are already there. When she carried again, it affected the second baby.
But shouldn't the incompatibility be there in the first case itself?
Incompatibility is there, but it did not cause damage because baby is delivered. So incompatibility was already there, but it doesn't cause the issue. It doesn't cause a problem.
But sir, blood is flowing from mother to baby anyway, so shouldn't it develop antibodies while the baby is in the womb itself? And
no.
Then
okay, I think what you're missing here, blood actually goes in one direction. Blood goes from mother to baby, never goes from baby to mother. So if baby's blood doesn't reach mother's blood, how does our immune system knows that there's Rh factor?
I was missing this point, I think.
Correct. Yes. So it goes in one direction. Yes. That is what you need to know here. Okay. So this is called incompatibility load. So we talked about several different types of loads here. Segregational, mutational load, recombination load, migration load. Okay. Right. This is about briefly what is genetic load and what are the different types of genetic loads. Now we'll talk about what is the method to calculate genetic load. Okay. And I wouldn't have talked about this, but I have seen a question in the exam on this. That is why I wanted to talk to you briefly about how is it calculated. Okay, 2013 question, 2013 and 2009. 2009, they asked this, what is genetic load and what influences it? What factors influence it? Okay, what is genetic load? On page number 63 from this book, questions under population genetics, you will notice that there were questions on genetic load twice. What is genetic load? What factors influence it? And a question came in 2013 where it asked for, what do you understand by genetic load in a population? So you explain this concept. What is genetic load and what is your understanding about it? How is genetic load measured? How is genetic load measured and what are the important factors that may influence it? Okay, so here we are addressing how is it measured? How is genetic load calculated? There is a formula for that. This is a formula, not too complicated. I'll explain you. So load, L is load. L refers to load. L equal to W optimal minus V divided by W optimal. So this is a formula. Load can be calculated using this formula. There are many, and it is some specific corrected, modified versions of this, but this is something really simple that you can understand and answer in the exam. Load equal to W optimal minus V divided by W optimal. So L is load here, and W optimal is the optimal or fittest genotype, because we're talking about it's a relative term. Genetic load is inferior to the fittest. An average, if people where there's optimal genotype, anything inferior to that can be considered to be causing genetic load. So here, what are we learning about? The fittest genetic genotype is W optimal. So WOPT or optimal refers to fittest genotype, the best genotype in that combination. And V or new, you can say new, refers to fitness, mean fitness, average fitness of the population. Excuse me. So this is average fitness, this is optimal fitness. So optimal fitness minus average fitness divided by optimal fitness or optimal genotype gives rise to load. So load is basically reduction in fitness, correct? So this formula can be used for it.
Is there a specific range for L, or is it like?
Yeah, I don't know. Yeah, it can be anywhere from 0 to 1. The range could be anywhere from 0 to 1. So see this, I'll explain this to you briefly. Yeah. If a population where W optimal is equal to mean fitness of the population, so that is V, mean fitness of the population is equal to the optimal genotype. So what is the load in that case? So average fitness of the population, the best genotype of the population is same. Then is there a load? No load, zero load. Because no negative effect of any change. The population is maintained at the best optimal status. So that only happens when there is no load. So L equal to 0. In the opposite case, mean fitness is drastically different from W optimal. So load can be as high as 1. 1 means 100%. So the value can be anywhere between 0 to 1.
Sir, here we are assuming that mean will be always positive. So that is what it is, right?
No. Mean doesn't have to be positive. So what do you mean by positive?
And if we take mean to be negative of, let's say, 1, so negative and negative, then.
Okay. No. How is it possible to have something that is negative here?
Negative is not possible.
Negative is not possible. Negative doesn't happen. How can you have a negative fitness? Negative means reduction, but how can you have a negative number for fitness? So negative is not possible. It has to be a countable number like this, 0 to 1. It has to be a positive value. Okay, I'll give you 1 or 2 scenarios to understand a little better, which I've given in the handout. Okay, so if all but one have a genotype of 0 fitness, all of them in all individuals except one have a genotype of 0 fitness. Okay, so V equal to 0 and L becomes 1. So 0 fitness is drastically affected. So load is highest, L equal to 1. So boundaries I'm talking about. 0 we talked about. Optimal phenotype is equal to the mean fitness. Best case scenario, 0 load. Worst case scenario, load equal to 1. Okay, so this is answer to your question, 0 to 1. It can be anywhere between 0 to 1. If a population with maximum possible fitness has a fitness of 1, then the genetic load due to a particular variant is, how do you calculate load in that case? Fitness is 1. The maximum fitness is 1, correct? So load can be calculated by L equal to 1 minus nu or W. Okay, negative. So meaning this is the optimal mean fitness of a population. So load can be calculated by the maximum fitness of a population minus average fitness of a population. So it's a difference between maximum fitness and the existing fitness. Correct? So this is another way of calculating load, provided you have some values given to you. So one is the maximum fitness. W minus is the optimal fitness. So 1 minus W minus is the load. So if you are given numbers like this, average fitness of a population, you can calculate. Let's say average fitness of the population is 0.4. What is the load? Average fitness of a population is 0.4.
0.6.
Exactly, 0.6. 1 minus 0.4 is 0.6. I'm talking about a very simplistic scenario, okay? But I don't think we need to get into the details about complex scenarios, but are you able to understand this?
Sir, for W optimum, so it is, what is the unit, sir? What is there a unit, or how do we say? We said it's a fittest genotype.
Yeah, I don't think there are units for this. They get cancelled out, right?
So what would be the
optimal
value?
minus V has same units divided by W optimal will have same. So they gets nullified. So these are just factors. So no units to it. Correct?
What I meant to ask is what would be, say, the value range of say W? Not that it matters, but I just
100%. We just say I think fold value. 100% fitness, 50% fitness compared to something.
okay.
This is a relative value, right?
So this is the fittest genotype in that particular population.
Exactly. Compared to, yes, existing in that particular population.
And it is compared to something other.
Exactly. You relate it. This is the fittest genotype. What about the genotype that you are studying or you observing? So this is just a relative number.
So probably it could be 1.
So maximum it could be 1, yes. Okay, right. There are more complex equations if you can digest. So this is one such equation. So if you have a mathematics background, you can definitely cite it, mention it, but I don't think it is necessary. But when I was going to literature, I saw these type of equations. So where they go little ahead of these calculations. So where they even calculate the frequency. Let's say there is a deleterious mutation that is causing negative impact. Okay, the frequency of that could be Q, like we calculated in Hardy-Weinberg equilibrium, P for one allele, Q for another allele. Like that Q value can be calculated. And if this reduces a population, an individual fitness by S, so reduction in the fitness is calculated by the value S. So what happens? In homozygous state, it will be H multiplied by S. Okay, remember we was briefly talking to you about inbreeding coefficient, like that coefficient for dominance can also be calculated. So that dominance coefficient multiplied by, fitness, reduction in fitness, that is S. So HS value can be used here. So where W negative can be 1 minus 2Q multiplied by 1 minus Q into HS minus SQ square, which will be a bit too much, right? So yes, there is much more to this concept, but we will have a limit how much we can understand on these components. So that's why I believe Probably if you talk about this L value and briefly explain how this can be different scenarios, two different scenarios of that, that should be sufficient, I feel. Okay, I'll put a little bit of more emphasis on the problems with genetic load estimation. There we may understand a bit about factors that can influence genetic load. Because I could not directly find information on what factors can influence genetic load. There are two possibilities here. One possibility is you talk about causes of genetic load, factors influencing genetic load. You focus on genetic load, causes of genetic load. Talk about mutation, talk about recombination, talk about segregation, talk about incompatibilities, like that, those components you talk about. So they could be factors that affect the genomic sequences, hence the genetic load of a population. This is one component. Second component, I noticed that when I was reading in literature, they briefly talked about issues with genetic load estimation. What is a problem in estimating genetic load? There are few assumptions that we have to make. So I'll briefly tell you about these assumptions. Okay. So if you can add some value to this answer of how genetic load is influenced and where are the issues in estimating genetic load, that will be great. Okay. Otherwise, you focus on the factors that influence them. The causes of genetic load could be the go-to area. Okay. So while genetic load seems like an intuitive measure of the burden of deleterious mutations, so it may sound, oh, we are able to calculate what is the effect on mean fitness of a population, or how much of fitness is reduced in a population. Correct? So it may sound as if it is actually giving a good measure of understanding the effect of maybe mutation, maybe recombination on the fitness of a population. But that may not be entirely true. First, the genetic load is a function of the parameters in a population genetic model. So meaning, actually, as we're talking about what is optimal, how do you know what is optimal? There need to be a model, there need to be an assumption related to that. So this is based on models. So the parameters that you consider here are created based on a model, and it is not something that can be directly or empirically measured from data. So you cannot directly pick. Sometimes it may be possible, but most of the times, given the complex interplay between our environment and our survival and even genes, it is difficult to pick that number. There is no quantifiable number that you can pick. So this is all made based on an assumption of models. So this is a best case scenario. This is how it's supposed to be. Okay, how does mean reduce as? So you estimate that number. It is not something that directly you can measure and calculate. So there is an inherent problem in calculating this. Okay, because it relies on genetic models. And genetic models are not necessarily the true representatives of a given population. It is in our best case, with scientists' best knowledge, they have created this. But it may not be entirely true. So we are relying on such models. So there is inherent problem in estimating genetic load. Okay, next. Thus, it is intrinsically difficult to work with. Okay, so intrinsically, we have a problem in calculating that exactly. Second, the computation of the genetic load rests on assumptions about the distribution of fitness effects and dominant coefficients. Also, what do we consider here? As I was telling you, we have to calculate the dominance coefficient. Okay, that is also an estimation. And also, we assume that the fitness may vary in a given population. That may or may not be true. Okay, definitely fitness has to be variable, but it has to move in one direction based on what alleles the population contains. Even if two populations have similar mean, so comparing two different populations, assume that they have similar mean derived allele frequencies. So meaning there are certain alleles, their frequencies result in a particular average mean value for which is, let's say, 0.5 for both populations A and B. Their genetic load may not be the same. So even if their fitness value derived from allelic frequencies are same, load may vary. So they're highly variable. So you cannot come to a clear conclusion or clear comparison between two different populations because they work on certain assumptions. And although it may sound like they have same load, but that may not be the case. Okay, the load could differ, is variable, if the distribution of fitness effects for segregating variants in one population is not the same as in the other. So we don't know what is the impact of segregation. So just merely calculating load will not give us the actual number, because inherently there is something else is going on in that population which cannot be calculated. So there is a problem. There is a problem in estimating the genetic load. Okay, so it's very difficult to compare and understand the populations. And this is just a brief estimation that we rely on to provide us some guidance in understanding the effect of mutations, recombinations, and segregations like that. Okay, so relevance of comparing a population with deleterious mutations to a theoretical population free of such mutation has been called into question, because sometimes what do? We talk about optimal genotype. What is optimal genotype? How do you know? So one thing that we just talked about, as I think Sunil or Vishal were talking about, is something that you can see in a population or something that you assume to be optimal. So you may have to start with an assumption that this could be an optimal phenotype, optimal genotype. Okay, but does that really exist is a question. So sometimes you have to theoretically assume that there is something called the best phenotype, but sometimes does it exist or not is a matter of question. So there is a problem in understanding the or calculating the genetic load. So this is a brief idea. This is a bit complex concept here. So just I want you to understand that calculation or estimation of genetic load relies on certain models, and also it relies on assumption that allelic frequencies of two populations, when they match, we assume that the fitness is also similar, but that may not be the case. Given the environment may be different for two populations, genetic load will also vary and the numbers may not be directly compared. So now, what factors does influence genetic load? What factors does influence the genetic load? So sources of these problems: environmental mutations, environmental mutagens, radiation exposure, exposure to drugs, exposure to chemicals, like where toxins, insecticides, pesticides, pollutants, okay, X-rays, UV rays, all these things could impact the source of variation. They can impact mutations. Genetic load is affected by exposure to all these components. So how mutagens? If you're into agriculture, you may be exposed to pesticides. You work in a chemical industry, you paint industry, you're exposed to certain chemicals, lead industry. These factors would influence. Okay, so it relies on those factors. Genetic load relies on those factors.
Again, these are same for mutations or any other variations.
Something that you can easily pick up is for mutations, right? So the problem is recombination. We don't have too much of control on recombination process. We don't understand very much about what factors control them. It's a random process. It varies from gamete to gamete, generation to generation. So we don't know exactly what factors influence. The easiest one here is the mutation. For compatibility load, you can say choice of mate selection basically. We don't normally look for blood groups, correct? In the biodata for when the biodata is created for marriages, I don't think we'll mention my blood group is this. Maybe nowadays it does, but usually not. So it is not one criteria for choosing the partner. Correct? There are certain factors like that, but mutations, we know that some exposures can create problems. Viral infections, various mutagens can create them. Okay. So next, as the frequency and homozygosity of recessive deleterious alleles increase, so homozygous condition is increasing. Selection can remove them. What normally the role of natural selection? If it is deleterious, selection will remove them, right? Supposed to remove them. Then from a population, reducing the genetic load. This is what is expected, right? Genetic load should not continue. At one point of time, the load coming from that particular sequence has to be removed because that is the job of natural selection, correct? At the same time, small population size relaxes selection against mildly harmful mutations, which may lead to accumulation of genetic load. But the selection may not act the same way in small populations. They are interbreeding. They keep adding that load. Natural selection cannot do much, as we discussed in genetic drift yesterday. Correct? So some harmful mutations are also maintained in small populations, thereby accumulating the genetic load. The efficiency of purging and the accumulation of mutations both depend on the rate of inbreeding. How are they maintained? What do they depend on? They depend on inbreeding process. There's more and more inbreeding happening, higher chance of bringing in homozygous recessive condition, like we were talking about in consanguineous marriages yesterday. Okay, so it's a balance, fine balance sometimes in maintenance of them. So completely dominant lethal mutations do not contribute to the genetic load. Why? They don't survive. People who have lethal genes in them, they will not survive till the next stage. They die off. Correct? In such cases, so natural selection has a role to play. Yeah, we are learning about the role of natural selection in maintaining or in purging the genetic. But some mutations still happen. We talked about this in lethal genes, right? In Mendelian inheritance unit. Why are some mutations maintained? Why are some deleterious alleles maintained in a population? Do you remember that? Why are some of these alleles maintained if they are lethal to us? Huntington's disease, we talked about as an example. Why is Huntington disease, although lethal, is maintained in a population? If the mutation is lethal, it should never exist. But why such lethal problems still exist in our populations?
Because they are conditional.
In a way, right? So what happens there? So I think we talked about sublethal, right? And what happens there? So they only express at later stages of life. So that is how they're maintained. So in the reproduction age, they don't have too much of negative impact on them. So they're maintained. Okay. So consanguineous marriages are inbreeding, increases homozygosity. In case of rare recessive deleterious alleles, it can increase genetic load. So inbreeding or consanguineous marriages also increase the genetic load. Okay, kind of correlates with what we already talked about. So you can also mention about consanguineous marriages and inbreeding, which increases genetic load because it brings in the two recessive alleles together.
So in the exam, is it enough to state these factors, or do we have to say elaborate a bit more?
It depends. Like for example, in the particular question I was telling you, there are multiple factors in that question. So there you don't have too much of room to expand on this, right? Mentioning them is sufficient. So in the particular question I was just mentioning to you, if they ask what is genetic load and factors influence it, there what do? You explain it. If they ask about many things, what do you understand by genetic load? How is it measured? What are the important factors that influence it? There you have to be really brief. Okay, depending on the demand of the question. So if you have to expand on it, what do you expand? How do you expand? As I was telling you, Huntington disease, you talk about Huntington disease. How Huntington disease, is maintained in a population. Second, you talk about consanguineous marriages. Discuss how consanguineous marriages happen and how they increase the genetic load. How, take example of sickle cell anemia, hemophilia, and explain how hemophilia condition or sickle cell anemia condition, or take Bradford project. Explain how in these communities the problem is increasing. Next, genetic drift. We also discussed this yesterday. It can influence genetic load. In extreme cases, deleterious alleles can reach a frequency of 1 in population because of genetic drift. Because accidentally, all those effective alleles are gone. So what happens? Those with the deleterious alleles may survive in the population. Inbreeding will result in more and more burden. So you explain with example. Talk about Amish populations. Okay, you expand them. because these units we have actually discussion happens in multiple different contexts in different units in this paper. So you can use them. That is why sometimes I say when you are writing the answer, your answer will be better after reading the another unit. Some of them actually strengthen your understanding. Okay, it will be really chaotic if I just pick up related content from different units. So as far as possible, I'm picking the order to meet to give you good understanding, but I cannot pick all the relevant topics from different units. So your overall understanding will be better once we finish the syllabus. Okay, but where possible, I'll just mention that, okay, you're going to learn about this in unit 12, in unit 9.4 in related to genetic counseling. But for now, just focus on what is genetic load, different types of genetic loads, how is it calculated, what factors influence it. So genetic drift, migration, consanguineous marriages, mutation rate, mutagens, they all can influence it. Okay, and this is one topic where I have not seen much sources in the standard literature. So this is something I found myself by reading articles. Okay, and some of the key references I have provided to you. The distribution of DILT is a genetic variation in human populations. A 2014 paper. An encyclopedia of genetics, genomics, and proteomics also talks briefly about genetic load. Okay, right. Short note, this is important. So as they then nicely asked sometimes, so what do you understand by it? How do you calculate it? What factors influence it? These are the things that we have seen in the past exams. Okay, right. So next we'll talk about last topic of this, genetic polymorphism. So what is genetic polymorphism? Have you heard this term, genetic polymorphism? Okay, if not, can you guess? Can you try to dissect the word polymorphism? Polymorphism.
Various forms.
Various forms. Poly means many, morphs means forms. Existing in various forms. In the context of what? In the context of genetic information, genetic polymorphism.
Genetic variety.
Enough genetic variety in a given population, because we're studying in population genetics. So enough variety in gene sequences of a population can be called genetic polymorphism. Correct? Okay, let's learn. So when is it called polymorphism? At what level does it exist? What are the different types of genetic polymorphism? And I've seen the questions have come on this part as well in the exam. The specific ones I'm going to talk to you. But first I'll give you an overview about genetic polymorphism, some key guidelines to understand this. Questions, if you notice in the exam, define genetic polymorphism, give details of its types with suitable examples. 2015 question, define genetic polymorphism, give details of its types and suitable examples. Similarly, before that, there was a question on write short note on genetic polymorphism. So explain genetic polymorphism, write a short note on it. And third time, the question was related to a specific type of genetic polymorphism called balanced genetic polymorphism. How is it maintained in a population? This is a topic, that example that we already discussed about. So I will explain you in what sense it is balanced genetic polymorphism. Okay.
So there is one more question in 2019 as well.
Correct, 2019. What is the question?
Differentiate between transient and balanced.
Correct.
Genetic.
So we are going to talk about
examples.
both balanced and transient genetic polymorphism as well. Okay. So this is the scope of this topic. So what is genetic polymorphism? Define it. Different types of genetic polymorphisms. Okay, there are two ways, different levels. At what level polymorphism happens, and what are the types of it? We are going to talk about both. Okay. So genetic polymorphism. Polymorphism in Greek refers to refers to many, morph refers to form. So many forms is polymorphism. So in other words, it is occurrence of more than one form in a given population. Occurrence of more than one form in a given population. Okay. So briefly, let's say that there are 100, let's say the population of 100, simplistic scenario. 99 of them carry the particular type of allele, one of them carry a different allele. Should there be a definition to say how common it has to be to call it or to consider it to be existing in a population? 1 out of 1,000, 1 out of 100, 1 out of 10,000. There should be a specific definition to that. Okay, so here they say a gene is called polymorphic if there is more than one allele is present at that locus, at least in 1% of the population, meaning 1 out of 100. If there is a new allele for that particular gene, then you call it polymorphic. If there is only A blood group everywhere and 1 in 10,000 people have it, can you call it polymorphic? 1 in 10,000 carries B blood group. So out of 10,000 people, 9,999 people carry A blood group, 1% carries B blood group. So can the blood group be polymorphic in that case? Yes, no.
No.
Yes. 1%, yes.
1 out of 1,000.
No.
No. It has to be 1 out of 100. So 10 out of 1,000. Okay. Or 100 out of 10,000. So that by rule, it has to be at least 1% of the population. Okay. And poly refers to many, so it has to be more than 2. Monomorphic means 1, 2 means dimorphic means 2, so poly means many. So A, B, O blood groups. It's a type of polymorphism. So that is why you always have to write occurrence of 2 or more clearly different phenotypes in a given population. And if they exist in more than 1% of the population, or the occurrence is more than 1%, it is called genetic polymorphism.
So, sir, if we have 1%, so 99% will be one form and 1% will be another form.
Come again? 1% and 99%. Yes. So 99 out of 100 people, 99 people are A blood group, one person is B blood. Okay, so it's dimorphism. The A, B, O. Okay, more than 2 such forms, refer it to as polymorphs. So variety, more variety in a given population, basically. Enough variety to match this rule, that is more than 1%, at least 1%. Okay. So I made a summary on this as well. I provided some introduction that you can read here, okay, later. Basically how common this is. So out of how many bases do you normally find genetic sequences being different? So read about that. This is general introduction to that. Okay, many of us actually carry some variety in our genetic information as per the Human Genome Project. As per the Human Genome Project, 99.99% of our DNA matches with us, with each of us. So we share about 99.99% of the DNA, leaving out only 0.1% differences. But even that 0.1 difference is sufficient, high enough, because our DNA, the letters in our DNA are huge. We carry 3 billion letters in our DNA. How many letters? We carry 3 billion letters. So 0.1% is approximately 30 lakhs. 30 lakhs. And within the 30 lakhs, we have enough differences. So inherently, we do carry enough differences in our DNA. So these differences with respect to particular sequence, particular locus, there may be multiples of them, multiple changes you may find in different people in a given population. So we are inherently polymorphic. There are enough regions in our DNA where differences in our DNA do exist. How, what level? We'll see. First, I'll talk to you about level of, at different levels. At what levels does polymorphism exist? Polymorphism exists at different levels. One is at cellular level, at the level of cell. Example is ABO blood groups. RBCs either carry A antigen, as I was talking to you already, B antigen, O antigen, RH factor. So, cellular level. The blood groups are examples of the polymorphism happening at the cellular level. So RBCs in different people carry different antigens. So at the level of cell, we are seeing polymorphism, correct? That is called cellular polymorphism. Okay, I want you to just be aware about the different levels of polymorphism. Yeah. Next, chromosome level. Polymorphism can exist at the chromosome level. We'll talk about this in unit 9.4. Structural abnormalities we see. Deletions, duplications to our chromosomes. Sometimes new sequences are added. Sometimes sequences are duplicated. Sometimes they are deleted, resulting in variations. I'll show you images, few images little later on this. So chromosomes can be different sometimes because either new pieces are added or some pieces are removed from the chromosomes. So two individuals' chromosomes sometimes may look different. So here the polymorphism is existing at what level? At chromosomal level. This rarely happens, but it does occur. We do have examples. What examples? We'll talk about that in chromosomal aberrations. I think briefly I'm going to talk to you and I'm going to show you a little bit of this in the next one. That's all in the next slide. Genetic level. So this is the nucleotide sequence level, letter level, molecular level. So our DNA sequence are different. The example I told you, 99.99% we are same, 0.1% we are different. At what level? At the DNA sequence level, our nucleotide sequences may differ between individuals. There are many examples I'm going to tell you for that. But yes, it can happen at cellular level, like in the case of ABO blood groups, chromosomal level, as the case with deleted, duplicated chromosomes. Genetic level, changes in our DNA sequences. And protein level, people do show different protein varieties. Hemoglobin, we're going to talk about in 9.1. Biochemical genetics, we are going to discuss this. People do carry different hemoglobins. Even in us, we carry different forms of hemoglobin at different stages of our life. So this can be called as protein polymorphism, changes in proteins like hemoglobin. Some people carry HbA, some people carry HbS. What are they? I'll tell you later. We're going to talk about this as a case study in biochemical genetics. But at protein level also, we do see differences among humans. Like that, polymorphism can exist at different levels. Cellular level to the sequence, DNA sequence level. Okay, let's now look into genetic polymorphism types. I'm going to talk to you briefly about the different types of genetic polymorphism. Single nucleotide polymorphism. It's one type of genetic polymorphism. It is single nucleotide polymorphism. Most common form of genetic polymorphism. Single nucleotide polymorphism or SNP. Very simple. Single nucleotide means difference in one nucleotide. So difference existing at one nucleotide. Let's say there is a gene of 1000 bases, 1000 base pairs. We are comparing between two individuals. 999 base pairs match. One base pair is different. So single nucleotide, difference in one nucleotide. So they are, what is SNPs? They are simplest and most common of all polymorphisms. There are many applications. We revisit this concept multiple times. They are single base changes or changes in one nucleotide. So as per the Human Genome Project, as I was telling you, 99.9% similarity that we talked about, 0.1% difference, as I was telling you, 80% of this 0.1 differences are SNPs, or single nucleotide polymorphisms. Okay, as an example for you to understand, let's say we have AAGCCTA. What is the difference between AAGCCTA and AAGCTTA? How many letters are different between these two sequences?
One letter.
One letter, right? This bold one, the one in bold. Instead of C, there is a T here. Like that, out of whole sequence, sometimes we may find, of course, it is not entire in the entire genome. At different locations, we may show one nucleotide change. Okay, which is the most common form of polymorphism. Okay, in this example, there are two alleles, one with C, this sequence, another one with T. So examples for single nucleotide polymorphisms. One letter change is very common in genetic diseases. Several genetic diseases we see are because of single letter change in that gene. Very good example is sickle cell anemia. You can pick up any genetic examples, any genetic diseases. Hemophilia, sickle cell anemia, Huntington's disease, cystic fibrosis. The examples we studied in lethal genes can be taken here. Examples we studied in lethal genes can be taken here. Okay, sickle cell anemia, cystic fibrosis are examples. Genetic diseases. So in case of genetic diseases, sometimes we come across what is called biological markers or genetic markers. Have you heard this term, biological markers or genetic markers? We have actually dedicated chapter on this, dedicated unit on this. Okay, 9.6, age, sex and population variations as genetic markers. So marker means a reference point. So what happens here? How do you know a person is carrying sickle cell mutation? One is seeing the outcome of the disease. People with sickle cell anemia will have anemia, so will have certain problems there. Hemophilia, blood clotting doesn't happen. So outcome can be seen. Otherwise, at molecular level, at the DNA level, by looking at that specific letter, one can identify, one can diagnose that a person is so-and-so person is suffering from sickle cell anemia, Huntington's, because you know that particular letter is different. But at one letter is different between individuals, so it is called single nucleotide polymorphism. Sometimes these sequences where variation is there and we know that variation exists and we can detect that variation, this region acts like a biological marker. It helps you to identify the problem. It is acting like a reference point, helping scientists locate genes that are associated with the disease. Many times these SNPs are associated with a disease condition, sickle cell anemia. You look for that letter, you find that letter in that particular person's DNA, you know the person is suffering from sickle cell anemia. So this becomes a tool to diagnose the problem. Okay, this is used as a reference point. It's like this, a landmark. If you want to understand this concept, a landmark. Let's say that you are trying to go to a particular place, particular institution, let's say, but you don't know where it is. You ask someone for landmark. They'll tell you, okay, this is by that so-and-so bank, by Canara Bank, by so-and-so theater, by so-and-so mall. So they're giving you reference point. You know where this point is, okay? And you know the institute that you are trying to go to is next to that mall, next to that theater. So what is acting like a reference point here? That particular location, that particular marker. Similarly, in our DNA also, there are markers. So they may not directly involve in the problem. But of course, that's not the case with sickle cell anemia. But otherwise, that is probably not involved in the problem, but that can guide you to the problem, guide you to the source of the problem. Because this source is identifiable. You can test it. You can do PCR test to check whether this particular sequence is present or not. We can use some probes to identify whether that sequence is present or not. If it is present, you know that person is suffering from so-and-so disease.
So
So
this
some
is
reference points act like markers.
so this is using the fact that 99.99% of our genome matches.
Yes, in a way, yes, but in different contexts, it does mean differently. Like, for example, in genetic disease, there may be differences exist among us, but we are looking for that particular change in our DNA. If you have that particular mutation in that particular gene location, yes, you know that this person is suffering from sickle cell anemia. And there you are not focusing on the rest of the variations. Similarly, in DNA fingerprinting, we do the same. DNA fingerprinting, we know that we are 99.99% similar, but if we look for those regions where we are different. So we use SNPs to identify the variable regions and personally identify so and so. Like you want to know whether you got a DNA from the crime scene. You want to know whether this DNA belongs to person A, person B, or person C. There is a genetic variation. The same variation may not be found in another random person. Only if that person happens to be there in the crime scene, this DNA will match. And you know that these SNPs have matched. And based on SNPs, you rule out the other people. You say this DNA doesn't belong to so and so person. It doesn't belong to B. It doesn't belong to C. It only belongs to A. How do you know? There is a method in DNA fingerprinting that you match the DNA with each other, with the DNA that is obtained from the crime scene. If it doesn't match, you know the reference point is not there. So that person is, the DNA doesn't belong to him. So he was not there in the crime scene. So like that, SNPs can be used in identifying them to a certain extent. You assume that 99.9% similarity is there, but we are more focusing on the variability, the 0.1% of the variability.
Yeah, but this is on the basis that how we will know to look for in that location.
This we figured out. We also have this topic in the syllabus. Genetic mapping, gene mapping we do. We identify where the location is. We can add the specificity. In the way you identify the sequence, you add that specificity. You are not just literally looking for, it's not like this. Like there is a 1,000-page book. You are not looking for, is there a letter E in this? You don't go like that. You go like this, like page number 3, paragraph 2, is there E? Second line, is there E? So you have to identify the location and then look for the presence of a specific sequence. So you really have to be specific there.
So it means that for most of us in that page, that para is same in a way. Matlab, we'll have that para there.
Either same or polymorphic. Polymorphic. Is it same letter? Because we have 4 possibilities. Either you have A, T, G, C. We can have any of these letters potential in that area. Okay, you will learn this. So we are just working on the theoretical basics yet, right? So technical components will go from unit 9.1. Okay, there I'll tell you how do you determine it. 9.1, that is why I didn't start 9.1 first. There we learn how to do it, how to identify the. Here we are learning theories. What is polymorphism? What level does polymorphism exist? How do we know it? How do we detect it? We learn it in unit 9.1. Okay, so that is why I was kept telling you the complete picture will only be obtained once we finish discussing all the units. They're all interrelated. If not all of them, or many of them. Okay, right. So this is briefly about single nucleotide polymorphism. There are many benefits of single nucleotide polymorphism. Nowadays, we are also using single nucleotide polymorphism. This is an applied area. This is important for DNA technology in medicine. So we are all using them in pharmacogenomics. So briefly, I'll tell you what is pharmacogenomics. So pharmacogenomics is the combination of 2 areas. One is pharmacology, another one is genomics. Pharmacology plus genomics is pharmacogenomics. Okay, so how do we use genomics in pharmacology? Pharmacology means study of drugs, right? Genomics means study of DNA sequences, genomic information. So nowadays, what are we doing? Based on our knowledge about genomic sequences, now we are hearing about this, right? I know the coronavirus strains DNA sequence or RNA sequence. I know the sequence of HIV. Nowadays, we have knowledge about the sequences. Okay. Similarly, we also have been sequencing human DNA as well. Nowadays, in India also, a lot of work is happening. We are sequencing humans' DNA, and now we have an understanding about where are these different sequences, where is the variable region in our DNA. So we have some understanding. So using that understanding to develop drugs that work better. So sometimes we refer to them as tailored drugs. We refer to them as tailored drugs or personalized drugs we call. Okay, and pharmacogenomics is one such area where we are using knowledge about DNA sequences to develop better drugs, drugs that work better. Otherwise, we use what is called one size fits all concept. Meaning, it's like this headache. You go to a medical shop, you say headache, more or less you'll get the same tablet. Of course, there are many companies, many varieties possible nowadays. But you get fever, you get paracetamol. Maybe different companies paracetamol they give you, but they'll give you paracetamol. Paracetamol is to reduce the fever. But not all drugs would work the same way on every individual, correct? So one size fits all approach may not work for all drugs, especially those that are complicated. What do I mean by that? Nowadays, in some developed countries, they're using pharmacogenomics for that, where people who are treated for HIV, people who are treated for cancers, Alzheimer's, not all drugs work in the same manner on every patient. So what do? They match the information about their DNA and how well the drug works. So meaning they're able to categorize people. Let's say people with certain sequence, certain SNPs may react well to drug A. People with a different sequences may react well to drug B. So because of this knowledge, what do? You match the information. You say, okay, if this person is carrying so-and-so SNP, so-and-so sequence, use drug A. If they're carrying a different sequence, use drug B. So you're able to effectively select the drugs which work better. So we are guided by the genetic information nowadays. So this can be called as tailored drugs, personalized drugs, and this comes under pharmacogenomics. And what is the connection to SNPs? Because SNPs are guiding us towards the direction. Okay, right. This is briefly about the very important form of genetic polymorphism, which is SNP, single nucleated polymorphism. Okay, understand these concepts. This is a handout I'm going to post to you after class. Okay, so is it clear what is SNP? Single nucleotide polymorphism, single letter change. Copy number variants. Sometimes we have changes in number of copies of genes. You may not have one gene for one character. Sometimes you may have same gene in multiple copies. Why is that? Why does it happen? Sometimes duplication happens. Sometimes same sequence gets duplicated. Whole sequence gets multiplied. Sometimes there are problems in the way we replicate DNA. So what happens because of that? Because of other reasons. Sometimes the DNA inside our body, genes inside our body, or DNA sequence inside our body, they amplify, they multiply, they duplicate, resulting in more number of same gene, resulting in variations in the number of copies of DNA segment inherited by individuals or carried by individuals. In some cases, major deletions removing entire genes can happen. So deletions, not only duplications, can take place. Thereby what happens? The removal of those genes can now read the information differently. It's like this. We actually have, let's say, CHAT chat. CHAT chat. H got deleted. It would read like cat. Is chat and cat are the same? No. So meaning of that sentence has changed. Meaning of that word has changed. So CHAT chat had become cat. Okay. So like that, sometimes deletions would change the meaning of that sequence. So two things we are learning here. One, copy number variations, duplications of same sequence. More copies you have. What is the impact if you have more copies of the same gene? What will happen? It's like this.
More of that protein, something is produced.
Exactly. So let's say it's simply, if you have to understand, instead of placing 1 spoon of sugar, you added 2 spoons of sugar. More dose, correct? Higher dose of sugar you will receive. Similarly, if you have more copies of the same gene, higher amount of protein, so it may become abnormal sometimes. That is what happens in cancers. We receive more and more growth signals even when it is not needed. So more growth signals, sometimes that happens. So like that, this is a variation. Variation in terms of copy number. Some people may carry 1 copy of the gene. Some people may carry multiple copies of the gene. In that level, it is called copy number variation. Okay. Next, very important one. I'm going to close in 5 minutes. Yeah. Restriction fragment length polymorphism. You read this, okay? In the material, you read this. So idea is this. You have to write down the idea. Duplication results in more copies of a gene. More copies sometimes means more protein. Can result in abnormality. But why is it polymorphism? Different individuals in a population may have variation in number of copies they carry. Okay, wherever you are not able to understand what I'm explaining, do stop me, okay? Yeah, 2 more types. Rflp, restriction fragment length polymorphism. Rflp, restriction fragment length polymorphism. Okay. So you all know what is restriction enzyme? What is restriction enzyme? Scissors, molecular scissors. Enzymes to cut DNA are called restriction enzymes. We learned about that in 9.1 in biotechnology. For now, just remember, there are some enzymes which can cleave the DNA sequences. Okay, some of these sequences are called restriction enzymes. Okay, to explain this, I'll give you a simple example. Okay, so it becomes easy for you. Okay, take a take a wire or take a rope. Take a rope. Put colors on the rope. Take a color pen or a paint. Put white marks, black marks, red marks. Okay. So you take a rope, you put white marks, 2 white dots, 2 black dots, 2 red dots, for example. Okay. So imagine that you also have scissors which cut at specific color. White scissors, red scissors, black scissors. Imagine that you took white scissors. How many times does white scissors cut if you have to only cut it where the white paint is, white mark is? How many times do you cut in this rope? There is a rope. There are 2 white dots, 2 black dots, 2 red dots. You took a scissor which only cuts at white regions. How many pieces does it make?
3.
3 pieces. How many times you cut?
2.
2 times. But it is not a loop. So basically you have a stretched wire, a stretched whatever. So cut it twice. So it results in 3 pieces. But let's say that you found another rope where there are 3 dots. How many pieces does that make if you cut it with the same restriction enzyme? 3 dots. 4. You cut 3 times, right? One piece is gone. Second piece is gone. Third piece is gone. Fourth piece also is formed. So 4 times. So what if you actually have here, the way I'm talking to you about the dots, 2 individuals. I talked to you about 2 ropes. Similarly, the 2 individuals in a population. One carries 2 sites for the same restriction enzyme. Another carries 3 sites for the same restriction enzyme. Will that result in different fragments when you use these enzymes to cut them? Such a polymorphism is called restriction fragment length polymorphism. Bit complicated, but actually logic is very simple. Did you understand the logic I explained you?
Here, are we assuming the length of the rope or DNA is the same and different number of points? Then we have different fragment lengths.
Yes, we are actually comparing same DNA in different individuals. Same region of the DNA we are comparing. In one case, you may have only 2 sites where restriction enzyme cuts. In another case, there are more than that, but for the same length. So what happens? Restriction fragment length polymorphism. Because you only cut it twice, you will have 3 fragments with 3 different sizes. If you cut it 4, you have 4 fragments with different sizes. So length of that fragment is varying, right? Variation is coming because of the site of restriction enzyme, site for restriction enzyme. Such a sequence, such polymorphism is called restriction fragment length polymorphism. Very, very applied area, applied tool in biotechnology. Okay, I'll explain that in the next class or in other sources, where when you cut it with the Rflp or enzymes like this, you know what fragment is where. And this can be used as a reference point. Sometimes the region that is cut may be related to genetic disease, because a change in the sequence either creates a mutant allele which causes disease. So by just performing some test like this, you know whether it is normal allele or diseased allele. It's like SNPs. This can be a part of SNP as well. SNPs can cause this. But here, how are you identifying it? You're identifying based on the length of those resultant fragments. That is why it's called restriction fragment length polymorphism. So read it. If you have a difficulty in understanding this, I'll explain that. Okay, the last one, which I kind of already talked to you, which is also a result of sometimes SNP, single nucleotide polymorphism, or more. So we have what is called short regions in our body. So it connects back to the variability in our sequences. We have 99.9 or 99.99% similarity, I was telling you, right? So 0.1% differences that we have. What regions do we have those differences? We have small regions called short tandem repeats. Short means small, few letters. Tandem means they repeat one after other. Example, we have ATGC. There is another ATGC, another ATGC, another ATGC next to each other. This is called short sequence, only 4 letters. and they are tandem, meaning they are next to each other, and repeats. They're repeating. ATGC is repeated, let's say 10 times. So in our body, in our DNA sequences, we do have short sequences which are present in tandem, meaning next to each other, and repeat multiple times. How many times do they repeat may vary. Let's say a person A has 10 ATGC sequences. Person A carries how many ATGC sequences? 10. Person B, let's say, carries 100 of them. So what is varying here between A and B?
Number of times they repeat.
Number of times they are repeating. You know, how is that called? Variable number tandem repeats. Variable number of times the sequence are repeating, because person A has 10 repeats, person B has 100 repeats. So variable number of tandem repeats. Tandem because they are next to each. ATGC is present next to each other. 10 of them are present. Another person, 100 of them are present. So this is called variable number tandem repeats. Okay, these are short tandem repeats. They are very small and their number may vary. So if number is varying, number of repeats are varying, you call them variable number tandem repeats. We'll come back to this sequence again because this is a principle behind DNA fingerprinting. And in unit 9.4 or unit 12, we are going to learn about DNA fingerprinting. Okay, right. So what did you learn here so far? Genetic polymorphism. What is genetic polymorphism? Poly means many, morphism means forms. At what is the minimum existence for this? At least 1% of the population should contain this variation. Then it is called polymorphism. At what level does polymorphism exist? Cellular level, like the case with ABO blood groups. Chromosomal level, like the case with the duplicated chromosomes or deleted chromosomes. Genetic level, as the case with the SNPs. Protein level, as the case with the hemoglobin variations. So it exists at different levels. Types of them we talked about. Single nucleotide polymorphism, the most common one, only one letter change at a given sequence. 80% of the differences among us belong to this category. Can be used in pharmacogenomics and disease diagnosis. Copy number variants, where number of same gene is repeated, multiplied couple of times. So variation in number of copies can be seen between different individuals in a population. Is there an impact? Sometimes more protein can be made because of this. Next, we talked about Rflp, restriction fragment length polymorphism. We have sequences where some enzymes called restriction enzymes cut. Some people may carry 2 such sequences, some may carry 3 such sequences in the same region. So the resultant here, when you add that enzyme, you may get 3 fragments or you may get 4 fragments, each with different length. That is called restriction fragment length polymorphism. So polymorphism is happening at the level of size of those fragments resulted because of cutting with the restriction enzymes. Very practical tool in population genetics. Also, it is commonly used. The last one is tandem repeat polymorphism. People may have repeat sequences and the repeat number of repeats they have may vary. That is why they are called variable number tandem repeats. Okay, the different forms of genetic polymorphism. Next class, I'm going to explain you the difference between balanced and a transient genetic polymorphisms, which has been asked in the exam. Balanced versus transient, and how is it different from what we talked about? This is the different levels. The next one is going to be one way of balancing the polymorphism. Okay, right. We're going to discuss that in the next class. And the next class will not be on Sunday. So Monday, I have to go to a hospital in Hyderabad. So most likely I'll be back by 3 o'clock. Okay, in case by 1 o'clock, I'll know if I'm not able to reach on time to home, or if I'm not able to reach some place where I can access internet, then I might cancel the class. But Sunday we will not have the class. Monday I'll inform. Otherwise, my plan is to take Monday to Friday, even Monday to Saturday. Okay, only when I'm not taking the class, I'll inform you. Otherwise, you assume that you have a class that day. Okay, so I'm planning to take class on every day, Monday to Friday for sure. If I miss one day, I'll try to take at least 5 classes a week. Okay, so assume, and I know sometimes I'm not able to reach on time to home. So I need to carry my laptop. Sometimes I don't. So I'm not able to give the information to you. If I don't announce anything, you assume that there is a class on that day. Okay, we'll go with that. Okay, Sunday no class. Monday most likely I'll take the class. If I'm not able to reach home on time, I'll let you know. Okay, right. Good. Any questions, guys? I'm going to post the material that we discussed today. Genetic polymorphism, genetic load will be posted to you right away. I'll post it. Okay, and go through the content. Okay, wherever you have difficulty, do let me know. Ask a question on Telegram group. Okay, I'll answer that question.
Okay.
Okay, good. All right. Vishal, were you trying to ask anything yesterday when I was closing the session?
So I was saying that I would also like to know that comparison that Yash asked, the difference.
Yes. Yeah, I forgot to deal with that. I think he asked about the difference between Darwinism and synthetic theory of evolution.
Synthetic.
So there, what do you do? Let me check that question if I understood correctly. But Yash is not there, right? Yes. So in UPSC, asked a question on how Darwinism differs from the synthetic theory of organic evolution. In handout, there are 3 differences are given. Will that be sufficient because it asks for a separate question? Yes, I think